Welcome, everyone. Glad you're here. I know you see me before I start, but the video doesn't. So, but people on the hello, welcome. All right. So anyway, today we're gonna have a little bit of fun. We're gonna be talking about genetics. We're gonna be talking about heredity. We're gonna talk about inheritance patterns, which is a lot of fun to go through and learn about. We're gonna do a lot of interactive stuff with the genetics. Inheritance patterns and kind of look at that. We're also going to look at very quickly mitosis, meiosis, although we covered the most difficult part of those concepts, which is meiosis, and talking about meiosis 2 and that kind of thing. You know, the splitting of the chromosomes. So we're going to talk more specifically about what the chromosomes are doing, how they're lining up, and all that stuff. And then we're going to finish off by talking about protein synthesis. So those are the things. Those are the three main things we're going to talk about today. So we're kind of jumping around, kind of moving around, and just keep in mind that we're pulling from actually. In your notes, I just caught that. That is kind of strange the way I put that, but it says Learning Plan 3, analysis of the role of DNA in controlling cell function and genetics. Underneath it says, oh, I get it. This is chapter 29, heredity, and chapter 3, the cells of living tissue. So that's where the information is coming from, but we start with chapter 29. That's why we see chapter 29 twice. I was confused. Why do I have that? So if you're not sure where to go for this information, I have the page numbers in there and all the figures and all that. So exactly where we are and what to focus on. So we're pulling a little bit from chapter 29, and then we'll focus on chapter 3 a little bit toward the end. But we're going to start off today talking about heredity. And this is kind of a fun thing to go through, because once you start to learn the different types of inheritance, you can kind of see how it relates to yourself. You can see how it relates to people in general. And what we learn in inheritance patterns or heredity is that most patterns of inheritance are not what maybe we've been led to believe is a common type of inheritance pattern, and that's the dominant recessive inheritance pattern. People say, okay, you have a mom who has blue eyes and a dad who has brown eyes. It's the brown eyes is dominant, and so therefore there's a better chance that the baby's going to have brown eyes versus blue eyes. Well, that's not exactly how it works, because eye color, hair color, and skin color, and a lot of different types of inheritance patterns actually don't fall under the strict dominant recessive inheritance pattern. Some do, some don't, but there are different types of inheritance, and we're going to talk about the different types of inheritance patterns so you understand how to do it. So we'll be working with the Punnett squares for some of these, but then when we get to some of the more complex types of inheritance, there is no Punnett square. It's just understanding that there are multiple things that control our genetic inheritance patterns. So for example, for skin color, eye color, hair color, a lot of those things, there's no way to predict what the offspring is going to have. You can just look at a range of possibilities. Does that make sense? Because you have to go back several different generations, and I'll show you examples of that. I won't get into that because it'll. All right, so let me go over and switch over to the PowerPoint so I can talk you through what we're focusing on here, and we'll cut to the chase. All right, so when we look at the chromosomes, say, well, what are chromosomes? And chromosomes are basically collections of DNA. And we know that DNA, hopefully by the time you get to this class, well, if you don't, that's fine. We learn again, right? So DNA is made up of pentose sugar base. We've got nucleotides, which are made up of amino acids, and they basically type out the code that tells your body essentially how to make you, how to form you, how to make more of you, how to recycle your worn out, how to rebuild parts of you that wear down, and so on and so forth. Also, if you have offspring, then some of your DNA gets copied and sent along to form the DNA structure of the offspring. We talked a little bit about that last learning plan when we were together. So, and we will discuss that again toward the end of today. But the DNA, for the most part, when your cell is not getting ready to replicate, the DNA is just kind of unwound and just hanging out in the nucleus of the cell. It's not until the cell is getting ready to replicate that the DNA will condense into what we see as chromosomes. So really the only time you're going to see chromosomes present is when the cell is involved in the cell replication process. Does that make sense? Otherwise, it's just chromatin. We call it chromatin. It's the genetic material that's just laying around in the cell. But when the cell does get ready to replicate and we need to package the DNA and organize it, what we're going to see is that the DNA is going to wrap itself around bunches of protein molecules called histones. Just showing them as like almost like little aspirin tablets or something like that. So if you picture like aspirin tablets, if you take 8 of them and you bunch them together for each group of 4, so there's 1, 2, 4, you wrap the DNA around that, and then you have another set of 4 underneath this one, and then you wrap the DNA around that twice. So that's why you see that the DNA wraps around twice. See that? And then what you do is you just keep wrapping it, and then it coils up. What does this coil kind of look like to you? I know we don't really have landline phones much anymore, right? But does it look like a phone cord? Do you think of a telephone cord? I got one in here, but I can't plug half of it. So, you know, if you think about a phone cord, so I can show you, demonstrate visually. All right, so behind me, you see the phone cord, and see if I take this phone cord and I push it together, you can see I can get quite a bit of. That's why the phone cord looks this way, because you can condense a long cord in a small amount. I can take this, I can stretch it out, I can make this quite long, right? So this is what it's doing. You're taking and wrapping it and coiling the DNA, and then once you get the DNA coiled, then what you do is you bunch it up like this. You kind of scrunch it up. So kind of scrunch the DNA, the cord together, kind of scrunching it up like that. Well, that's what happens. It scrunches up, scrunches up, scrunches up. Just gotta learn how to plug things in. So what you see then, picture that scrunched up phone cord. That's what's happening over here toward the right side of the screen. What you're doing is you're scrunching that up. See how you're weaving it back and forth, back and forth, back and forth, back and forth. And as you do that, and you kind of make these loops with the coils, and then you loop the coils, that is going to form chromosome. So if you picture that you've got the DNA wound twice around the histones, then you coil it up like a phone cord, and then you loop it, push it all together, you can just begin to comprehend how much DNA and how much genetic coding goes into one single chromosome. It's a lot. It's a lot of information. But think about creating a whole new living organism and what instructions you would need to do that. I mean, it's a very long list of things you have to how to make and do, right? So it's a significant amount of information. So in our cells, in human cells, as you see in the notes, it says that we have what's called a diploid number of chromosomes, consisting of 23 pairs of homologous chromosomes. Okay, so what they're showing here are all the chromosomes that are laid out. When they lay them out like this, it's called a karyotype. Now, these don't look like chromosomes, because chromosomes kind of look like an X, and these just look like, they look like single lines. That's because the the arms and legs, so to speak, of the chromosome are so close together. Can you picture that? That's why it doesn't look much like a chromosome over here, for example. Okay, so this is 1, 2 chromosomes. You see that? Okay, so if we count those up, we should have 46 pairs of chromosomes. Now, what they do is they arrange the chromosomes. They don't line up like this. This is computer. This is done by a computer. They say, okay, well, this is chromosome 1, this is chromosome 2, this is chromosome 3. And what they do is they line up the pairs together, and they call those pairs homologous pairs. Homologous pairs. Okay, so now I know that this is not how genetic patterns work, okay, but I just want you to play with me a little bit so you understand how this works. So let's say that the genetic coding for brown eyes is on chromosome 1. Now I can't verify that that is or isn't. I'm just making up a story, okay? So imagine that the genetic coding for hair or for eye color is on chromosome 1. All right? You follow me? Okay. So you received one set of chromosomes from mom, one set of chromosomes for dad, right? So on mom's chromosome number 1, you received the genetic coding for eye color from mom. And on chromosome number 1 from dad, you received the eye color coding or genetics for from dad, eye color for dad. You got it? Okay, so this one here is mom's. This one here is the one you got from dad. You got it? Okay, so homologous pairs are chromosomes that carry the same genetic information on the respective chromosome. All right, so that's what it is. So chromosome 1 carries genetic information for XYZ, and you get XYZ from mom, and you get XYZ from dad. And then how they play together is what makes you who you are, what genes are quote unquote switched on or off, okay, if you want to think of it that way, all right? Or combined. We're going to talk about that, the different inheritance patterns, all right? So if you line them all up, chromosomes 1 through 23, there you go. But then at the end, the 23rd pair, these are a little bit different. These are called sex chromosomes. And the reason why they call these sex chromosomes is because the person's gender is determined by the chromosomes that are here in this last pair. Now, because these are 2 chromosomes that look about the same size and they're rather large, I'm going to go out on a limb and say that this is a karyotype for a female, because females have 2 X chromosomes. XX, when it comes to the sex chromosomes, equals female. A Y chromosome, as they call it, then it's a male. But the Y chromosome, as we'll see in a little while, Y chromosome is smaller than males. That's why you can't get mad at men for being men, because right? That's our excuse. See, he's like so on the missing from the Y chromosome is the ability to ask for directions, to put the toilet seat down, to stop playing with the remote control. I mean, these are all the things that you miss because you don't have the Y chromosome, right? See, so you can't get angry at men because we lack the genetics. We're genetically inferior. All right, we're inadequate. We're inadequate. All right. So anyway,
You gotta go.
what's that?
Never mind.
So is the honeymoon over already? Or you? Oh, I was gonna say, experienced problems already? My goodness.
Go home and tell them. Figured it out.
Figured out.
Figured it out.
So now we know the secrets of the world, right? Why men are just. Yeah. All right. So anyway, the other 22 pairs are called autosomes. So the first 20. That horrible. The pen is not horrible. They're called autosomes. That's what they're called. Yeah, that's the thing. You know, it's like a jingle. Again, there's autosome. Autosomes, right? Not even he can help you now. All right. So anyway, so here we go. So we got the autosomes. We got 23 pairs of chromosomes. 22 are the autosomes. The 23rd pair are the sex chromosomes. Now, the genetic information that's found on these chromosomes are called alleles. The fancy word for traits. Follow me? Like the trait for eye color. Or I should say eye color is the trait, right? So where you see the genetic coding for each trait, that's called an allele. So the allele for thickness of hair, the allele for alleles for eye color, the allele for whether you have attached or not earlobes, that kind of thing. You follow me? So the allele is the trait that you're looking for, or that you're observing, or that you're measuring. All right. So as we move on and we talk about dominant and recessive alleles, we're talking about dominant versus recessive traits, if that helps you with the translation. Okay. So an allele is sort of like a trait. So as it says in the bottom of page 1, alleles are any two matched genes at the same locus on homologous chromosomes. All right. So what we're seeing here, we're going to introduce you to some terminology. If the two alleles that control a trait are the same, the genotype is homozygous. All right. So for example, if we look at the alleles for hair color, okay, now this is oversimplified. This is not how hair color and eye color is necessarily controlled. They're just simplifying this, okay? But let's say that on this location, on this homologous pair of chromosomes, look at the color coding down here. So this is dad's chromosome, mom's chromosome. So on this homologous pair of chromosomes, at this location, you will find genetic coding for hair color. Now you can see on dad that he has, that we're showing two lowercase h's, which is our way of noting the blonde hair allele. You can make up whatever you want, okay? If you want to do X, it can be X. If you want to do B, it doesn't matter what the letter is, just as long as you, whether it's capital or lowercase. And this isn't necessarily dominant recessive. It's just lowercase h is the allele that was the symbol we're going to use for the blonde hair allele, and the uppercase H is the symbol we're going to use for the brown hair allele. Got it? This isn't dominant recessive necessarily. It's just the symbols that we're using. You follow me?
So
That up.
oh, okay.
Okay? What I'm just trying to illustrate to you here is the terminology. So if we see that we have two of the same symbols for the allele on the together, okay, so we have two lowercase h's, we call that homozygous. Okay, so it's the same allele. So if we have two uppercase h's, would that be homozygous also? Yes, it would. Also homozygous, because the letters match. If we had, instead of a lowercase h here, we had an uppercase here, then we have uppercase and lower. Now the alleles are different. So we would call that heterozygous. Okay, so they're different alleles, non-same alleles. Right? Now, when we do talk about the dominant recessive, yes, we do have some alleles that are dominant, and what that means is that it masks, at the bottom of page 1, masks or suppresses the expression of the other allele, and usually is indicated by a capital letter, and likewise, the recessive allele is suppressed by the dominant and will only be expressed in what we call the homozygous recessive condition. We'll talk about that. So okay, we'll get the terminology. Okay, so just you understand homozygous is the same allele for both, and heterozygous is different alleles? You understand that, you're okay where we are right now. Does anyone not understand that yet? Okay, all right. We don't have to worry about inheritance patterns or anything yet, okay? Now, one of the things that helps us out as a species, and any species, is genetic variety. We don't want to have children that are exactly the same as the parents, genetically. And as a species, you want variation, you want variety, because it makes the species stronger and better adapted to survive. So just through the process of meiosis, which we'll talk about, you see genetic variation, but it's completely random. And we're going to see how it's randomized. So what we'll see is that the way that the chromosomes line up is completely random. But there also is randomness in what happens when the chromosomes line up together. So during a process of meiosis, you will see the homologous pairs of chromosomes line up next to each other before the cell gets ready to divide. And what can happen sometimes is that we see something called crossover, where the legs or the limbs of the chromosomes actually cross with each other and they exchange genetic information. So now you get these kind of like hybrid or newly formed chromosomes with a little bit of dad on one and more, a little bit of mom on the same one. So now what we did is we kind of crossed over, and some of dads ended up with some of moms, and some of moms ended up with dads. And so now when these chromosomes, through the process of meiosis 2, when they go their separate ways, this one goes into, for example, one sperm, and this one will go into a different sperm, or this will become the polar body, and this one stays behind, depending on which gender, which parent you're in. Now you can see that we've sent off a chromosome that's different than the original, right? So there's genetic variation that it can occur very randomly. Now you can see, instead of the homologous pair of alleles, now you can see we have heterozygotes. So what we're trying to get at is it's completely random. And we get at that here, and that's what this picture is trying to show you, is that even if you have only 3 chromosome pairs that you are lining up, and how many pairs are we actually lining up here in meiosis? How many pairs are we lining up? 23, right? Yeah, 23. So this is only 3. And look at the variation. Go this side gets this, and this side gets this, okay? That could happen. Or, depending on how they line up, you could get this. So we have 1, 2, 3, 4 different scenarios, and we have more, but there are 4 different scenarios that we can come up with very easily for just 3 pairs of chromosomes. Imagine 23 pairs. So there's a lot of variety. But it's kind of funny how it works, because you put mom and dad's chromosomes together, and maybe you've seen this. Some kids look a lot like one parent, and not much like the other. Or some kids don't look like either parent, but they look the same as each other, right? You can see that all the kids look identical to each other almost. That you can say, well, I can tell they're brothers and sisters. So it's very interesting how that works, but we don't have to spend much time on it. But just wanted to kind of hit on that. So we'll talk more about meiosis when we get to that, because this is chapter 29, and they're hitting on a concept, not realizing that we're going to be hitting it while we're here together. All right, so the first term is the term Punnett, and we're going to do some Punnett square stuff today. We're going to learn how to use Punnett squares and how they work. Let's just talk about how we put things on a Punnett square and what we're getting at. This book does something I haven't seen in any other book, and I don't understand why they do, but they put the Punnett square kind of on an angle like this. You don't like it?
No.
I don't care for it either, but whatever. I guess they're trying to do it so it shows equality, that one isn't going on the top one. I don't know, whatever. Fine, whatever. Okay, sure. All right, that's what you need to do. All right, so anyway, so if we look at the gametes, not the gamete. Okay, let's say that we've got the mom's genes and we've got the dad's genes. Okay, so when we look at we go through meiosis 1, then we go through meiosis 2, we end up with separate gametes for each one. So what they're showing us, for example, is that we're going to inherit maybe this one from mom, and then maybe we'll inherit this one from dad. I don't know. So what you do is you take the alleles from each of the parents for whatever trait it is that you're looking for, and you split it up. So here we've got the heterozygous combination of some sort of trait that we're looking at, right? Capital T, lowercase t, so we take the capital T, put it here, put the lowercase t here, because there's a 50-50 shot. The offspring may inherit this one, or the offspring may inherit this one from mom, okay? It's a 50-50 shot. We don't know. If this is the genetic coding for dad, and we go through meiosis, one sperm is going to end up with the dominant, I shouldn't say dominant. One's going to end up with this allele, and the other sperm is going to end up with this allele. Might want to look at over here is this may be the genetic information that sticks around. This might be the polar body down here, or vice versa. This might be the one that sticks around. This might be the polar body. So what you're doing is you're just playing with probabilities. It's like, flipping a coin or rolling a die. You have the same chances no matter how many times you roll, but sometimes you roll a 6 3 times in a row. You roll a different number every time you roll, okay? Or you flip a coin, you're going to flip a coin, and while you have the same chances, sometimes you can flip heads 3 times in a row, right? So what you do is you just combine them, put the uppercase T's together, and here we've got the lowercase t with the uppercase T. We've got the lowercase t with the lowercase t from here, uppercase T with the lower. So you just combine them, and then what you do is you look at the probability. So you can see that there's a 1 out of 4 chance that it's going to be, if this were dominant recessive, if it were, you would say homozygous dominant. They have a 50% chance that offspring is heterozygous for that trait, and then you have a 5% chance that it's okay. We'll play with this a little bit. All right. So when it comes to dominant recessive inheritance, I want you to understand that dominant doesn't always mean quote unquote normal or expected. Okay. So on each of us, unless I really am missing something, each of us has 5 fingers on each hand, right? And you'll be surprised, some people don't. I was at a store one time, and this woman was paying for her product, whatever, at the checkout, and she had an extra pinky on her, extra finger on her hand. I'm like, well, that's something you don't see every day. But actually, what you see here is, where's it now? Red digits.
Normal digits.
Where's a? Oh, no, that's not it. But anyway, for example, it's not on here. But for example, the dominant gene, or the dominant gene, the dominant allele is actually for more than 5 digits. The recessive actually is normal number. Same thing with webbed digits. Recessive inheritance pattern is normal digits. So if you inherit the dominant allele, then you'll have webbed digits. Dwarfism is another one. Dwarfism is actually a dominant allele. Most of us are of quote unquote normal stature because we've inherited the homozygous recessive inheritance pattern of what we call normal bone formation, which keeps us. So you have to differentiate. So normal doesn't always mean dominant. Dominant doesn't mean normal. So just want to point that out to you. So some things might be interesting to you that you see here, like freckles is dominant versus absence of freckles. I don't know what it is. Widow's peak, straight hairline. What if you have no hairline, right? Get into that. Actually, we'll talk about it. Balding. All right. So anyway, this is the dominant recessive inheritance. So actually, there is a somewhat finite number of traits that are inherited through dominant recessive. And that's where we're running the problems when you have, and I don't think it happens all that often, especially nowadays. But you hear the horror stories of a kid going to school and then they do Punnett square, and they're assuming that the inheritance pattern is dominant recessive, and then the kid goes home crying because there's no way I can be my father's daughter because my dad has blonde hair and I have brown hair, and there's no way I can ever have blonde hair because, all that kind of stuff. You just have to be very careful. All right, so let's play with this. Do you guys have a piece of paper? Does everyone have a piece of paper? Something to write with? If you don't have a piece of paper or something to write with, then raise your hand. I have paper here. So let's do this together, okay? We're just going to do a simple dominant recessive inheritance pattern. All right, so go ahead and make your Punnett square, just like this that you see on the screen here. Then I'll tell you how to work through this. Have their square made, square, and then just put the cross in the middle. All right, so what we're looking at here is we're looking at cystic fibrosis. Okay, so dominant, and so cystic fibrosis, or the inheritance of cystic fibrosis, or the presence of cystic fibrosis, is passed on through dominant recessive inheritance. Believe it or not. So this one's pretty straightforward. Okay, so let's just get our symbols set here. Capital C means no cystic fibrosis. Lowercase c, fibrosis. So capital C means the allele for no cystic fibrosis. Lowercase c is the allele for cystic fibrosis. You might want to make your C's the big C is really big, and then the lowercase c is really small, so that way you can tell the difference. Okay, the father and mother are both heterozygous. Okay, so how are we going to put the alleles on this Punnett square if both parents are heterozygous? What does heterozygous mean? One of each. So father. So up here it says father's genes. So what are we going to put at the top above these squares? Yep, we're going to put a big C, and a little. Okay, good. Easy enough. Mother is heterozygous, so that means what? Same thing, one of each. So next to one of the C's, you put a large C, and next to the other box, you put a lowercase c. Okay, put these together. Oh. Okay, so yours look like this up here. We have one that has homozygous dominant, two that are heterozygous, and then one is homozygous recessive. You have that? That's what they have. Okay, so I want to introduce you to two terms down here because these are going to be important for you to understand. Okay, so we have the genotype. Genotype refers to your genetics. Okay? Phenotype means physical expression, right? So let's differentiate for here. So the genotype, what percent genotype? So what will we say? 25%. We have a 25% chance that the genotype will be dominant. I'm sorry, homozygous dominant, right? homozygous dominant, right? Okay. What about this one? What percent? 50% is what? Yep, heterozygous. Okay. And then what about the remaining 25%? Okay, so those are the genotypes. So if these two parents have an offspring, 25% chance that the child will inherit the homozygous dominant alleles or allele pattern, 50% chance that the child will have the heterozygous genetic coding just like the parents, and 25% would be homozygous recessive. All right, but now let's look at the phenotype. Physical expression. What is actually going to happen? So when we have two capital C's here, what does that mean?
They won't.
Right, so, okay, so they won't have it here, okay, so 25% no cystic fibrosis, okay, good. What about these two? 50% chance, I'm hearing maybe 50% chance. What are we really seeing here? This is dominant recessive. And if capital C is no cystic fibrosis and that suppresses this one, what does that mean? Is this child going to have cystic fibrosis? No. So 50% no cystic fibrosis, but they carry it. Yep. Right, but they carry the trait or the allele for cystic fibrosis, just like the parents did, right?
So
This one here?
no,
Okay, so the heterozygous?
the heterozygous one,
Yes.
the big C. Have it. Is the dominant trait always going to go over the recessive trait when there's
Yes, in a dominant recessive inheritance pattern, whenever you see the dominant and the recessive allele together as the genotype,
right?
the phenotype is that the dominant will always suppress the recessive, always.
Okay.
So whenever you see the heterozygous arrangement, in this case, because the dominant is no cystic fibrosis, they will not have cystic fibrosis. But their offspring could, yes, which is what we're going to see here with the remaining 25%. So the last 25%, what does that mean? They have it, yes. They have cystic fibrosis. So you could have two parents that don't have cystic fibrosis, yet have an offspring that has cystic
Right, because
fibrosis.
neither of those parents had it.
The parents have
They were
cystic fibrosis.
heterozygous.
Correct. So you could have two parents that don't have cystic fibrosis, but have a child that has cystic fibrosis. Very real life. You could have two parents that don't have cystic fibrosis, and the child has cystic fibrosis. Where does this come from? Well, it came from both the parents. Each of the parents had the heterozygous alleles. So then it just was that 1 out of 4 shot that. So is it possible that you could have these two parents have 4 children and none of the children have cystic fibrosis?
Sure.
Absolutely. Is it possible that they could have 4 children and all 4 have cystic fibrosis? Yeah, possible. Very unlikely, but this is what you do. This is what genetic counseling kind of does. They look at the inheritance patterns and they say, okay, based on your genotype and your spouse's genotype or the other parent's genotype, these are the percent chances that your child could have. Concern for some parents because if they have a certain genetic condition that is in the family, they want to know what the chances are of their child having it, so they can make an informed decision as far as whether they want to have children.
Everyone in my family but 4.
Yeah, that's exactly it. It's exactly that, because if you look at this one right here. So the recessive, so everybody in your family can,
Yeah, only
except for
four,
one.
4 people can.
4 people can and one can't?
Everyone can, like, there's like 30 people.
Yes.
Like 4 people
Okay, so you're saying 30 people in your family can roll their tongue, but 4 people can't.
can.
Because it's a dominant recessive trait, that means that the parents of these offspring all have the heterozygous
Okay.
pattern. Because the only way you can end up with a non-tongue roller is if one of those 4 boxes is so this would be the dominant, I'm sorry, the homozygous recessive. So that means that the parents carry the trait for the inability to roll the tongue, but because they carry also the dominant allele, they can roll their tongue. But they have children that can't, because the children, you have 2 heterozygous alleled parents that have children and then end up with that. Yeah, so the parents are carriers. Yeah, but this one's more, harmless. When you consider other things that could be. Oh,
No, I
maybe you are constipated when you try.
I don't think
Okay, before we take a break, I'm just going to take you through this one, and you don't have to do this one with me if you don't want to. This is one that I have fun talking about because it relates to me. I just like talking about myself. You can tell that, right? I just love talking. Okay. Anyway, no, I don't. But so here's the thing. My dad is bald. I'll show you a picture of my dad soon. My dad is bald, and but when I was growing up as a young boy, one of the worst things you could ever be told is that you're going to be bald, right? You don't want to hear that. Right. So you tell a young guy, you know what, you're probably going to lose your hair. They don't like to hear it. So it's like the stages of grieving. High at first, and you bark. I love being bald because I don't have to, because my hair is like, you know what Josh Groban is? Afro, it was crazy. So I actually love it. I used to wish that I wouldn't have such thick hair, and it's amazing when you wish, pray and pray and pray. I just did a lot of praying, and first delivers in a big way. Anyway, so you hear this thing. Well, if your mom's dad has a full head of hair, then you don't have to worry, right? If your mom's dad has a full head of hair, even if your dad's bald, you don't have to worry about it. That's total BS. If your dad's bald, you might be bald. Okay, it doesn't mean you will, and that's because baldness or male pattern baldness is what we call sex influenced. Which that means is that the inheritance patterns affect males differently than females. Okay, so if you look at these alleles. A capital B is bald. A lowercase b is not bald. See, in females, okay, so I'm trying to figure out how I have this. So if a male inherits these allele pairs, either homozygous dominant or the heterozygous, the male will be bald. But women will only have baldness, and it's not even as severe as men, but women will only experience baldness when they inherit the homozygous dominant trait. If a woman inherits the heterozygous alleles, she will not be bald. But if a male inherits that same genotype, the male will be bald. Okay, so it affects men and women differently. So I'll share it. So this is my family. We're a variety of bunch. We go the whole gamut. I'm the second youngest. I have another sister who this is my younger brother. Got lots of hair. He's got no
You
problem.
can tell it's going.
I'm the second youngest. Well, it's pretty thick. It's just he's got all the dreadlocks and all that stuff. Here's me. You see me. I'm the second youngest. I have a sister. She's not bald. She's not in this picture. Here's my older sister, my other older sister. She's 6. Not bald. Here's my dad. Bald. Then I got my oldest brother here. He's 10 years older. So when it comes to my brothers, you look at my brothers for the most part, even if the variation is, I have the least amount of hair of all my siblings. Dad, right? So you tell yourself, that if your father's mother has a full head of hair, you'll be okay. So these are my dad's parents. Father, bald. Okay? And my dad looks a lot like him nowadays. And I found out just a couple of years ago that the number of years between myself and my dad is the same number of years my dad and his dad are apart. So that was it. Here's my mom's dad. My mom was from Puerto Rico, so you can see the nice tropical weather and full head of hair. Now, he was young in this picture, but he never lost his hair. He lost his eyesight. He lost his legs because he was diabetic, but he didn't lose his hair. So this is how it works out. Because I'm bald, but I have brothers who are not bald, and my sisters are not bald, I was able to kind of work backwards and figure out that these are the alleles of my parents. So my dad, his genotype looks like this. And my mom's genotype. Now, here's the one that is the tough one to figure out right here. Is this an uppercase B or a lowercase b? So was my mom also heterozygous, or was she really homozygous recessive? Well, the answer comes into what the males look like in our family who are bald, okay? So first of all, I don't have any bald sisters, so that's a big clue. But also when men inherit the homozygous dominant, like they're like bald as cue balls. I mean, they're like bald, like all hair is gone. I mean, I still have a little fuzz up here. If I inherited this, I mean, there'd be like no hair up here at all. I mean, I'd be like smooth as a cue ball. They call that frank baldness. It's like severe baldness. And nobody in my family has frank, severe baldness. You looked at my grandpa, he still had a little bit of fuzz up here. My dad still has a little bit of fuzz. I still have a little bit of fuzz. My brothers, even if they're losing their hair, they have some hair, they're kind of losing it. They still got it. So nobody in my family has frank baldness, and I don't have any bald sisters. So my best guess is that my mom's alleles were the homozygous recessive. And if you play this out, now you can ask the question, if I have it here. So last thing before the break. So if you ask the question, okay, if you are a male in my family, what are the chances that you'll be bald?
50%.
50%, right? Because if you have this, then you're bald. If you have this, you're not. So if you're a male in my family, yes, you have a 50% chance of being bald. What about if you're a female in my family? What are your chances of being bald? 0. Because if the woman inherits this, now she can carry it. So could my sisters potentially have offspring who will potentially be bald?
Sure.
Yeah, if they're males, yeah, big time, and I do. So I'm just waiting. Everybody joins me. Baldism is that even a word? Baldness. All right, here we go. We'll see, because I have all nephews. All my siblings that had children, they all had boys. Curse of the bald is coming together. I'll have company in my misery. I'm the only one out of all my siblings that has girls. I have only 2 children, and both of them are girls. All my other siblings have boys. So, yes, the curse of the heterozygous baldness trait coming to you. So we'll see. But so far they seem like they're pretty good. I think actually, honestly, I think all of them will be all right.
Is there-
Usually you'll start seeing it as early as what you said, you know, for guys, it depends on how far along they are in their maturity, the progression of puberty. So if some boys enter puberty earlier, it could affect them earlier. It has to do with testosterone levels. Now I entered puberty very early. I mean, my voice had changed by the time I was in 6th grade. But I didn't start going bald noticeably until I started to notice it when I was about 9. I had a friend. All depends. But yeah, that's just something to think of. And so this is where you know, we're talking a little bit after class last week about how things apply to things outside the classroom. And we're going to look at more things. There are ways that you can look at inheritance patterns. As long as you know what the inheritance patterns are, you can start to play around with it and kind of do things like this and guess at what your parents' alleles were or might have been based on you and what you are. Or you look at your own children and you say, oh, you know, and I'll show you the next one after the break when we talk about like curliness of hair. And you can look at your own children and say, oh, that's interesting. I can see now why that is. Why my children have, you know, the kind of hair they have. So yeah, so I just wanted to share that with you. It's an interesting thing to look at. But this is what we call dominant recessive inheritance, but it's a sex influenced dominant recessive inheritance. So that's because the gender of the individual influences the actual phenotype. The genotype could be the same, but the gender affects the phenotype, the presentation of it. All right? Ready for a break? Okay, let's go ahead and take a break and then we'll come back and we'll look at a couple other ones. Right. Okay, so there's another type of inheritance pattern that we look at. This is called incomplete dominance. This is a little bit, this is kind of like dominant recessive, but we also call it co-dominance. So if you got a piece of paper, go ahead and make your Punnett square, and we'll work on this one together. And we'll tell you how.
And what did you call the baldness one? Was that dominant?
That's a dominant recessive inheritance, but it's sex influenced. But still dominant recessive. Great for baldness that will. Press the recessive. One with the alleles. Capital C is curly hair. Lowercase c is straight. We're looking at here. Father. Now we gotta figure this out. What? Now father has wavy hair. Mother. How do you think we get wavy hair? Have an allele for straight hair. Got it. Heterozygous. Mother both have wavy hair. So walk me through. What do you have for the father's alleles? What would you put? Thing. Oh. So what are the genotypes? 25% what? 25% is homozygous.
Oh, homozygous.
Well, I guess you would just say homozygous. There's no dominant. Okay. Homozygous for, yeah, just homozygous. Okay. So then, zygous for the allele for curly hair, you have 25% homozygous with the allele, right? Heterozygous, you got it. Now, going to the phenotype, as you were saying, Joey. 25% is what?
Homozygous dominant.
What?
Curly hair.
Yeah, 25% curly hair. And what? 50% weight. Percent. Great. Yeah. Wavy hair. And then you get a child, they have a child that has straight hair. Well, there's curlier hair than that. So if you look at your hair, curly, wavy, straight, parents' hair, kind of see how that works. This is what we call incomplete dominance or co-dominance. So neither one is going to dominate over the other or suppress the other. They're both expressed together. My wife has very straight hair. I have wavy hair, so my daughters kind of have in between. All right, this next one is what we call multiple allele inheritance. That you can have the same phenotype with a combination of several different types of genotypes. how this works. What page is this table on? 11 what?
1100.
1100. Okay, so make sure you have that open. Types for the Punnett square. So go ahead and make a Punnett square. Well, what is that? Okay, so before we can do this, we need to look at the available alleles. Father's going to have type A blood. Or type A. Okay. What's the genotype for type A?
Or no?
Yep, that's one. Well,
Oh,
it's
that's
not
for
two
all.
lowercase i's. It would be uppercase I, A. Yeah, right?
Or,
Or
up.
so this I stands for. I'll just give you that so that you kind of know what. Isoantigen. So you think about blood types. Blood type B, type O, AB, no blood group. So an antigen or an isoantigen, they call it isoantigen, but you can just shorten it. Antigen or isoantigen is like a name tag. You put it on, hello, I'm type A. Or if I have A and B, I'm AB. If I have no name tag, I'm type O. I'm a 0. Okay? Got it? So when you see IA, that means that on for each of the alleles or the possible gametes, you're carrying the genetic coding for that A antigen. So we can just put father is type A. So we do, instead of doing the capital IA, I'll just put A. Okay, but we have the other one we can do. Okay, now for the mother, she has type B. So what are the possible genotypes for B? Very similar. IB, IB, IB. Let's start with mom and we'll just do. Okay. So if we did this, what would we get for blood types for the offspring? They would all be AB, type A. Okay. That's one possible. Okay. Let's just switch around the moms. Let's do this one now. Carry an antigen. Now what do we have? Type A, yep. Then, are you catching it?
Mhm.
Everyone get So what I did now is I put the lowercase I for each of the parents. Now, what are the types? What are some other types we see that we didn't see before? Type B blood, yeah. So the answer is, what are the possible types what are the possible blood types of the offspring? All of them. All of them. But it depends on the parents' so if you know your blood type and you had a child with someone else, you know their blood type, you can guess at what the offspring's blood type is. From your book, this is shows you possible combinations. My wife and I, she has O and I have AB. Daughters, so my daughters either are type A or type B. But I know that their alleles would either be mom is type O, right? So that would have to be the way. So if, like I said, if you know your blood type and the blood type of partner that you had children with, approximate, depending on your blood types. I mean, if, like I said, if it's A and B, well, then you know it could be anything. It all depends. All right, so that's multiple allele. So you can you can take two parents who have type A and type B, but A and B can be expressed in multiple ways, multiple. All right, now we're going to get into what we call sex-linked, not sex-influenced traits, or sex-linked traits. And here's a great picture. It shows you how few, how little genetic information is carried on the Y chromosome. This becomes, I wouldn't say problematic, but it just becomes an issue when we look at sex-linked inheritance. So this is what happens. We get a certain trait that is not carried on the sex chromosomes, but there's only so much room on the Y chromosome bus. It's small. There's only so much room on it. So if there is a certain trait that's carried on the X chromosome of the male, it's a quote unquote undesirable, and it might not be undesirable, just is a trait, there's nothing on the other X, or there's nothing on the other chromosome because it's a Y to cancel that out or negate it. So what happens is it has no choice but to be expressed. So if you want to put together another Punnett square, let's do what we call sex-linked inheritance. And I'll walk you through this because this one's a little bit different than the ones we had done before. Because we have to use X and Y chromosomes because we're talking about sex-linked inheritance. So when you are doing these problems or thinking about it, you have to know what kind of inheritance pattern you're working with. If you don't know what kind of inheritance pattern you're working with, it's hard to set up the Punnett square and do it correctly. So you will know that for this one, we are doing sex-linked inheritance. So what we first have to do is we have to put down the father's sex chromosomes. So a male has what combination of sex chromosomes?
X,
X and Y. Okay, so we have to put the X down here and we have to put the Y down first. Okay, how about the mother? What does she have? X, X. Great. Okay, good. Now what we do is now we look over here at our code or our symbols. So a plus would be normal vision, which we're going to treat kind of as a dominant allele. Okay, so this, in some ways, these sex-linked traits are kind of like dominant recessive. Okay, and then we have when I say dominant recessive, I mean normally passed on and normally expressed versus not, okay? And then the colorblind, for simplicity's sake, we'll just kind of treat it as just so you have that set up. Okay, we're gonna go over here now. So if a father is colorblind and a mother has normal vision, but she's a carrier, what would be the genotypes or phenotypes of the offspring? Y, X, X. So the plus was what, normal vision, right?
Right.
Now what we do is we kind of treat this, the plus and circle as like the alleles. So the father is colorblind, okay? Now here's the thing. There is no space here on the Y chromosome for the trait for colorblind or no colorblind. Color or whatever you want to call it, there's no room on the Y. So it only is found on the X chromosome. So the trait for normal vision or colorblind vision, those traits are only present on the X chromosome. So the father is colorblind. So what will we use? The O or the plus?
It'd be XO.
Yeah, we have X, and I'll just put a superscript here. Know that the father is colorblind. The mother has normal vision, but she's a carrier. So what will we put for hers?
X plus.
Plus, right? X. Is everyone following me on that? Good? No? Okay. So, color trait, what are you confused on? Let's start there. I lose you.
So the Y would,
So we have the X chromosome here, we got the Y chromosome here. The X chromosome is physically larger,
That's probably.
Y chromosome is physically smaller, right? Can't carry as much genetic information on a Y chromosome as you could on the X. So the trait for these sex-linked inheritance patterns, color blindness being one of them, color the allele for color or normal vision only be carried on the X chromosome. There's only room on the X chromosome, not be carried on a Y. There's not enough room. Does that make sense? So when we're putting this on the Punnett square, you can never put a plus or a minus here. I mean, a plus or a circle here. We can't do it because there's no, the Y does not carry that allele.
It doesn't have.
Not enough room.
Okay.
So it only can be carried on the X. The male has an X chromosome. Know that the father is colorblind, then we know that he is carrying that colorblind allele. Certain traits,
Okay.
yeah. So you would know ahead of time that it's a sex-linked inheritance pattern, and then that's how you figure it out. Okay? Back with us then? Okay, so the mother, she's not colorblind, but she's a carrier. So we know that on one of the X chromosomes, she has the normal vision allele. The other one, she's a carrier, so she's a color, so she does carry the colorblind allele. Okay? Good now? Okay. So when we put this out here, we're going to have two X's, right? XX.
Yep.
That makes sense because there's a 50-50. Offspring will be male and a 50-50. Right? I don't know, whatever. I didn't say 50% female, 50% female. Oh, but it. So anyway, 50% chance of male, 50% chance female. Yeah. So now what we have to do is we just have to carry down the alleles. So we have a plus and a circle, right? Here. Follow me? Okay. And then here we've got circles, The X from here is this. We don't have anything on the Y. We don't have anything on the Y here, nothing here. And then there's nothing here. Okay. So, what are the genotypes, phenotypes of the offspring? Okay, so if we have, looking at the females, what's the genotype? 50% heterozygous, right? Of females, so the female will be a 50% chance heterozygous, and a 50% chance that she's homozygous with the colorblind allele, okay? For the male, 50% chance that the male has the allele for normal vision. And so now when we look at the phenotypes, what do we see? If you're a female, you have what? What would be the phenotype? 10% chance you are.
Or carrier.
Yep, well, either one. You're right in both. So 50% chance that you would be a colorblind female, or a 50% chance that you would be a normal vision female, but you carry the colorblind trait. And over here, if you're a male, what does it mean? 50% chance that you are
Normal vision.
normal vision, yep, and a 50% chance that you're. Well, it is 100% chance that you're colorblind if you inherit this pattern.
Okay.
But if you're a female, you have a 50% chance of. Got it? So you have to differentiate between the males and the females because it's different. So the reason why I show this is because some people are led to believe that it's impossible for women to be colorblind, but it's not. It's very rare. You would have to have a colorblind father who has a child with either a colorblind female or a female that is not colorblind and is a carrier. Possible. Just doesn't happen all that often. Because most of the time, this is what. Most of the time, this is what you see. Oopsie, you don't see that. Most of the time, what you'll see is you'll have a father not colorblind. Child, not colorblind. Well, it usually happens. The reason why the boy is colorblind is because the mom was a carrier. Where'd the mom get that from? Nope.
Yeah.
Exactly. I'll take it back so we see it, but this is just what I'm showing you here. Okay, so if we go back a generation, and look at her. Looking at her, this is what happened. What happened a generation back? Father, colorblind. Father was colorblind. Yep, so the daughter was a carrier. So then mother, carried that allele, this one. Then chance now that when you have a colorblind father, that the daughter will be a carrier. Maybe higher if the mom is also a carrier. But then you would end up with a colorblind female, don't you? So most of the time it's a mother who doesn't carry any colorblind, normal vision. The father's colorblind. He has a daughter. That daughter has normal vision. Then that daughter has a child, and the child is colorblind. It's a boy. That's when you start to see things that skip generations. So that's why you see grandpa was colorblind. Oh yeah, and look, his grandson is colorblind. And then grandson has a daughter, normal vision. Now the daughter has a child, and she has a boy. Look, just like grandpa, colorblind. See? So this is where some things seem to appear to skip generations, because it's only carried on the X chromosome. Sex-linked inheritance. If you're a male who's colorblind, yes, hope you don't have grandsons. Well, but it could be if the woman you have a child with, you could have a colorblind offspring, male or female. Yeah. So it's just kind of fun to play around with. You know, if you know anybody, like I know people in my family who are colorblind, you know, and not colorblind, and you can see it skipping. Wife's grandpa was colorblind. Mom is. My wife's brother is colorblind. My wife has cousins. We're also colorblind. Daughters, both with normal vision. Daughters had sons, colorblind, yeah. So, okay, the rest of the inheritance patterns outside of the ones we've talked about are usually what we call complex inheritance. As it says in your notes, page 3, letter E, polygene inheritance, which means that there are multiple alleles that are all factoring in types of phenotypes. So what they're trying to show you here on this picture is that it's very possible. Let's say you have two parents, a couple generations back. One parent had very light skin, dark skin. They had a child that had an intermediate, kind of in-between skin. If they have an offspring with another person with a similar background that has intermediate skin color, but their parents also, one was very light and one was very dark, what it's showing you is that when these two parents who have an intermediate skin color have a child, they could have anywhere from a very light-skinned child to a very dark-skinned child. Now this one you just say, oh, well, that makes a lot of sense because grandma has really light skin. Grandpa has really dark skin, so I can see how, you know, little Billy, light skin, whatever, okay? But what we're trying to show you here is that the highest probability is that the children are going to have probably about the same skin color. Where you see some children who have darker skin than their parents. And you're like, where the heck did that come from? It came from the generations above. And if you go far enough back in the history of that family, you'll probably find someone who had color skin that this darker skin offspring has.
Sorry. Brothers. Look more like my brothers than his.
Yeah.
And same mother, same father.
Yep, same mother, same father. Yep. Just because you go back far enough, you'll find that. Yeah. Especially in Puerto Rico because you have people coming.
Darker than all of them. But
Yeah.
I mean, same parents.
Right. Same parents.
Two that are, real light, and then
They're darker.
darker.
complexion.
She's, dark. She looks Dominican.
Yeah, there's a lot of variety. Especially a place like Puerto Rico, and especially there because you got people backgrounds and so forth coming to that island.
I mean,
But
like you
there's
said,
so-
the
such a
same,
back-
same parents, you know,
Yeah.
same
yeah.
grandparents, same parents.
Yep. Same thing goes for like eye color, hair color, and so forth. So if you have a certain hair color and your spouse had, or the spouse or person, partner, which you're having children with, whatever, has a different hair color, sometimes the hair color of the offspring will be just like mom's or just like dad's or somewhere in between. Or maybe it comes out of nowhere. You see that. You have two parents who have relatively dark hair and they have a child that has blonde hair. Hanging out with the milkman. No, you just gotta go back far enough and you find the great grandma's hanging out with the milkman. That's a discussion for another day, right? So what you're gonna see is that most of the inheritance patterns, to be honest, are more along these lines. Eye color, hair color, skin color, those types of things are, and things beyond color. I mean, a lot of things are controlled by what we call polygene inheritance. Okay. Are we all hereditaried out? I am. That's enough to remember, right? So the key is just use your Punnett squares, know what kind of inheritance pattern you're working with, practice on your Punnett squares. You got. All right, let's change things up a little bit. We're going to move on to cell growth and reproduction. I'm just going to talk through a couple things, a couple of important points about cell division and the cell cycle itself. All right, so when a cell is going through its life cycle or its normal cell cycle, it's going to go through two main stages. We're going to go through what we call the phase that's called interphase. Have the mitotic phase, which is the dividing phase, all right? So interphase and the mitotic phase. Now, one thing I want to make very clear to you and remind you of, and if you know this, you'll never go wrong with cell division. Always remember that there is this stage, S stage. This is the growth and DNA synthesis stage. Important to remember. What's going to happen here is you have a single cell. During the growth phase, it just gets larger, okay? But during this growth and DNA synthesis phase, go from 46 chromosomes. Okay? So when you complete the S phase, how many chromosomes do you have?
92.
92. And then the final growth is you get a little bit bigger, and then we start condensing the chromosomes and all that stuff because we're getting ready to divide. We'll have how many chromosomes? Yes. So when you start mitosis, or when you start meiosis, how many chromosomes do you have? 46. So remember, whenever we start mitosis, or whenever we start meiosis, we have twice the normal number of chromosomes. We always start with 92. Start with 92. And if you do that, you'll never go wrong. You'll never mess up. Your second term is the term growth. I'll take you through it. Where it'll really screw you up is like mitosis, you can usually figure out, you know, oh yeah. We have two cells that have 46 because we had the cell divided and you get that. But where you really can get screwed up is if you try to go through meiosis, meiosis with this. So what's happening here during the replication of DNA? If you need to know, what you do is you take the DNA, an enzyme that's going to basically unzip and unwind the helix. You're going to break it apart, and then what's going to happen is you're going to use an enzyme called DNA polymerase, which is going to match half of the DNA ladder, so to speak, up with its corresponding nuclear partner, okay? And you do that both ways, so you end up with 2 strands of DNA that were identical to the original one, okay? Both. Or meiosis, depending on which one you're going through. Sense?
Whenever you start.
So what you're seeing here is that at the end of your cell cycle, let's say we're just doing mitosis. Up with one cell and another cell. Each of those cells has 46 chromosomes in it, okay? I'm not going to worry about one of them now. We're just going to worry about one of the pair that we created. That one cell will get a little larger, and then we'll double the chromosomes. So 92, then it gets a little bit larger, and then we split it in half, and now we have 46. Meiosis, what happens is, again, we do start, we don't start from a divided pair. We just start with a parent cell. Parent cell that has 46 chromosomes. We have to duplicate the chromosomes first. Then we start splitting them, and then we split them. Meiosis, you're splitting 92 chromosomes twice. Give you 23 then. So you go from 92, you split that in half, you go to 46, you split 46. I'll take you through it. I'll show you in just a little bit. So when you start mitosis or you start meiosis, you always, because this is where you start, right here. Right here is where you start. You'll always have 92. Okay? Follow me at least for now?
Yeah.
Okay. We'll go through it. I'll take you through it. So this is what we're gonna see in mitosis. Before we get into the cell dividing readiness phase, like I said before, the DNA is just unwound, it's hanging out in the nucleus, okay? So when we start getting ready to divide, the DNA is going to condense into chromosomes. How many chromosomes are in here? Start of mitosis, how many do we have?
92.
Yep, 92 chromosomes. We have 92 chromosomes in the cell. We have 2 sets of 46, identical, okay? So what happens is the chromosomes start to arrange themselves line. We have all 96 chromosomes. They line up along what we call the metaphase plate. File. Very organized, very structured. Very, very structured. As opposed to meiosis, which is unstructured and random. So the next step is anaphase. Anaphase, you will pull apart those chromosomes. So how many go this way? How many go this way? 92, how many go this way? On this side, how many go on this side? Yep. So you have 46 and 46. Now we're going to get 2 daughter cells. How many chromosomes? Right. So when we enter the mitotic phase, think of a dog. Think of a dog that's being potty trained. It's going to pee. Dog to pee on a mat. Okay. Prophase. Metaphase. But before we start to pee on the mat, we have 92 chromosomes, remember? Okay. Always remember, you always start with 92 chromosomes. 92 chromosomes, and then you go through the prophase, metaphase, anaphase, telophase. You'll divide those 92, and you'll get 46 chromosomes on each side. Okay with mitosis. Mitosis is pretty straightforward.
92.
Letters full, yes. Yep, after telophase, we're at 46 in each daughter cell. Yep. You got it. So if this is how we grow larger. We take one cell, this is like what we talked about when we were in learning plan 2, where you had the single zygote, and then it divided into 2 daughter cells, and that became 4, and then 8. So we got to the morula. Okay, so it's growing larger. Well, it didn't grow larger because it didn't go through the growth phase. So it's going through synthesis, so it was doubling the chromosomes and splitting, doubling the chromosomes and splitting. It wasn't growing larger. It was just sharing the cytoplasm. Not in your book, okay, so you're going to have to just you can just draw the picture. Okay, but this is meiosis. Meiosis has 2 stages, meiosis 1 and meiosis 2. Meiosis 2 is a lot like mitosis. So once you get to meiosis 2, you can pretty much treat it like you did mitosis, and it's pretty straightforward. Okay, a little difference, but we'll go through it. But meiosis 1 is the one that's different. So we start with a parent cell. Now we have the PMAT here too. We have prophase, but it's prophase 1, metaphase 1, anaphase 1, and telophase 1. So because we're in meiosis 1, we give it a 1. Okay, so at the start of prophase 1, how many chromosomes are in here? Very good, 92. Wonderful. Okay, now what's going to happen though is, remember in mitosis, we saw that the chromosomes line up single file. So picture this room. If I had all of you line up single file along the middle of this room, and then I'd rip you in half. That's what we do. We're going to do something different in meiosis 1. I'm going to say, okay, line up in the middle, but line up in pairs. So you're going to pair up with a person. So if you imagine an imaginary line down the middle of this room, one person's on one side of the line, and then their homologous pair is on the other side. Does that make sense? So we're lining up according to homologous pairs. So dad's chromosomes and mom's chromosomes are going to line up, but it's random, okay? So maybe we have dad's over here and mom's here, and then maybe mom's is here and dad's is here, and then dad's is here and mom's is here, and dad's is here and mom's is here. It doesn't really matter. The chromosomes line up along the middle. That's what they're showing right here. How many chromosomes? 92. So we have 46 pairs lined up along the middle, okay? That's metaphase 1. Anaphase, anaphase. Anaphase is where we pull them apart. So during anaphase, half the chromosomes are going to go this way, half the chromosomes are going to go this way. Random, because we don't know how they lined up, right? Maybe moms were on one side. Maybe it was really organized. Maybe all moms were on one side and all dads are on one side. But the chances of that are very slim. And remember, we had the crossover. So even if they're lining up, we still have the chance for crossover, where we're combining and mixing up. Right here. Okay. But anyway, by the time we are done with meiosis 1, we have 1, 2 cells. How many chromosomes in each? 46 each. Yes. Are these identical to each other? Probably not. No, right? Because of what?
Because it was.
Because it was randomized, okay?
Now,
So,
once.
go ahead.
Okay, so those pairs, I know the pairs match up randomly,
Yes?
but then when they'd still be random.
It'd still be random because they just say, line up, quick, line up. Everybody lines up with their homologous pair and then they go. So it could be, it's completely random. So now we have 2 cells, each with 46 chromosomes, but they're non-identical already, okay? So they're not the same as So now we have two non-identical cells, each have how many chromosomes in them? Now it's just like mitosis, pretty much. So now all the chromosomes line up, single file down the middle. You rip them apart, and so now how many do you have in each of these? 46, you split in half, now you have
23.
20. That we have 23. So if we look at mitosis, we have one parent cell divides into how many daughters? 2 daughters. How many chromosomes in each? Nope.
No, 40.
As meiosis, on the other hand, we start with one parent cell. What do we end up with? How many daughter cells? 1, 2, 4. So we have 4 daughter cells. How many chromosomes each?
20.
Non-identical, right? Right? If I say so? I don't want, if I say so though, I want you to understand it. Do you understand it, or are you still confused? Are you
Because
confused
they
at how we
all
got to
just-
4? Math working out for you? Do you understand how you get 4 with 23 chromosomes? Is that where you're stuck? Let me do a blank slide and we'll do it together. How about that?
So mitosis starts first.
Sort of.
All cells go through both?
Tell you what, let's take a break. Kind of clear your mind. And then what we'll do is, when we come back from the break, I'll explain to you, I'll give you the big picture. I'll explain how we do the whole, I'll give you the whole 9 yards, okay? I'll give you the whole thing. But I want you to get up, move around, clear your mind a little bit, just, do whatever. Come back, and when we come back, I'll give you the whole shebang. Then you'll understand how everything fits and where it all belongs. Okay? Make sense?
Yes.
Okay, good. We'll do that. I want you to blow your brains out here. Okay, so as promised, I'm gonna the whole shebang here. Help you
Yeah.
figure out where everything goes and how everything fits together, which is not always easy. Okay, I'm gonna present a couple different scenarios. Okay, so let's do just scenario, oh, I gotta turn my pen. So, let's do scenario. All right, let's do scenario one. So scenario one is the easiest. Okay, so that's gonna be, you have, skin cell, okay? Now the epidermis is made up of a but not, we're not going that, we're not zooming in that far. Okay, so we've got the epidermis. So the epidermis is made up of one of the layers is a basal layer of cells called the stratum basal. You might remember this. And the stratum basal is a mitotic cell layer. So what happens is this cell divides off, and the cell that divides off moves up. So in order to do that, we need to have 92 chromosomes, right? We split in half, 46 stays down here, and 46 goes here. So then the new cell that moves up has original parent cell with 46 chromosomes. Now, again, we'll go through the same thing. It'll go through this synthesis phase so that it'll eventually bump back up to 92. It'll split off again and give off another 46. 46 stays down here, and then it'll bump up to 92, and then it'll split again. Do you see the pattern? Or am I completely losing you here? Follow me or not?
Yeah.
Good? Everybody's good? You're good? Okay, we're all good. So that's mitosis. Start with 46 chromosomes. You go through the S phase, which bumps up the number to 92 chromosomes. Then that splits, divides off into two identical daughter cells, 46, 46. That's mitosis. And I think you all know that, but I just want to make sure I present this so you understand it. Okay, now, let's look at scenario number 2. We're in the testes of the male. Whether you want to be there or not, we're going to go there. All right, so in the testes of the male, very similar situation. We have testes. We're living in the testes, all right. So we've got these cells within the testes. And what they're going to do, start with 46 chromosomes. Guess what they're going to do? They're going to go through the growth phase. So they get how many? 92. Split. So you get 46. This bottom part, this bottom cell, I'm just going to bring it over here. This bottom cell now is going to stay here, and it's going to do what it normally does, which is grows and gets larger. This cell that comes off of here identical to each other right now? They underwent mitosis, right? Just like they did in the basal layer of the skin, or the epidermis. So are these identical?
Yes.
They're identical, yes. This cell stays down here. This cell now will become the sperm cells. Okay. So, how I would explain this then is that this will go through its replication again, 92. Okay. Now we begin meiosis 1 with this 92. Down here is going to go up to 92 again, but then 46 are going to stay down here and then process. You follow me? So the sperm cells, the cells in the sperm are going through mitosis first, like you were asking, and then one of those cells is going to stay back and continue to do mitosis. The other one is going to begin meiosis. Does that make sense? Everyone following me? Now this one is going to begin meiosis, okay? You have a cell that has 92 chromosomes. What happens? Chromosomes are going to line up like this, right? Then what's going to happen? Metaphase. Then they're going to split. So half the chromosomes go this way, half the chromosomes go this way. So when we get the, all right, so we have how many chromosomes in each of these now? 92, so we have 46. So this is the end of meiosis 1. Now remember, I'm still talking about sperm cell here, okay? So now the next step is meiosis 2, where we take the chromosomes, the 46 chromosomes, we line them up in the middle, okay? And then we split it, right? Meiosis, end of meiosis 1. So now meiosis 2 is where we take these cells with 46 chromosomes each. They're not identical. This is not identical to this one. You follow me?
Mhm.
Okay, so we split these now. Have 1, 2, 3, 4 sperm. With, got it? Okay. So we start with mitosis to give us these first 2 cells. Each one of these cells is then going to go through the synthesis phase. This one continues to do mitosis. This one will enter meiosis. Got it?
They're not identical once they complete meiosis.
Correct. When they do this,
They're not identical.
this is not identical to this one. Correct. Not identical. They are not identical. So by the time you finish meiosis 1, you have 2 cells, each with 46 chromosomes, but they are not identical.
What about
Nope.
Those are, so we have
Four sperm cells, 4 gametes that are not identical to each other. You got it. Good with that. Scenario number 2. We're good? Scenario number 3. Now we're going to the ovary. Or a woman is born. She's given all the eggs that she will ever have in ovary. Okay? Underdeveloped egg cells. One of them. So, that underdeveloped egg, then when she reaches puberty, her ovaries become active. One by one, the body will select one of these eggs, develop and grow. One that gets picked. You are the chosen one. You are going to be released from this ovary, and good luck. All right. So when this happens, now it's going to enter the synthesis phase. So the 46 is going to become how many chromosomes? 92. You got it. So 46 gets bumped up to 92. Okay. And larger. We're going to split this in half. You're going to have 46 on this side, 46 on this side. 46 sticks around. The other 46 gets expelled as the first body. So we got rid of 46, so how many do we have left here? 46, okay? So let's clear away all the garbage here. Chosen one, because we got rid of the first polar body, now has 46 chromosomes in it, okay? Now that 46 chromosomed egg ovulates, it gets released from the ovary, travels, starts its way down the uterine tube. About a third of the way down the uterine tube, it meets Mr. Sperm. Sperm fertilizes this egg, and we said before the fertilization can take place, the egg has to complete meiosis. Remember? Meiosis 2. Why do we have to undergo meiosis 2?
23
So you can only have
chromosomes.
20.
Yes, we have 46 here, so we have to get rid of 23 as the second body. Are eliminated. Left with what?
Egg never,
It did. Right here.
oh, here,
The
okay.
first polar body. That was
Gotcha.
meiosis 1.
So it never underwent, well, it did undergo mitosis.
Well,
Or
no.
no, it
No,
never
it didn't.
really underwent mitosis.
It went through the synthesis phase. Doubled the number of chromosomes. Once it was chosen, you're the chosen one, then it doubles the chromosomes to 92. Splits those. That's the first polar body. And when it leaves and gets ovulated, if it gets fertilized, then it discards the second polar body. So for the female, she discards 3/4 of the chromosomes. Only uses 1/4 of them. Whereas the sperm, you end up using all of it. 92 is split 4 ways. You get 4 sperms. Male, it's all about quantity. You want as many sperm as possible to give you the best chance. Whereas in the female, you get one egg that gets pampered and brought up and expelled with hope that that's the one that's going. Is that helpful? You understand now how it kind of all fits together?
So then the
First polar body would be reabsorbed by the body, and same thing for the second.
Chromosomes.
Yes, then you'll combine the 23 from the mom and add. They don't necessarily line up again in homologous pairs until they would undergo meiosis again. So the only time that you see the 23 pairs actually marching up and lining up together as a pair is when you're preparing to divide for meiosis. Otherwise, in mitosis, they're lining up single file. Meiosis, lining up mom, dad, dad, mom, dad, mom, mom, dad, just randomly, but they split. Yeah, meiosis, yes. Feel better about that now, at least understanding big picture, how it all works. It depends on the gender. But if you think about, yeah, if you look at it from a purely numbers point of view, look at meiosis, start with 92, that to 46. Non-identical, non-identical because we had the chromosomes line up like that, So for the males, it's really easy because you can just say these become 4 sperm cells. Females, it's tough because this one is the first polar body, leaving this behind. And then this becomes the second polar body, leaving this number behind. That's the difference. Sperm use all the available chromosomes. As far as the egg, it's going to discard 75% because it only needs 23 to merge with the 23 for the sperm. Better about that? You okay to move on? Right. I took you through, didn't I take you through first day class, like a shortened version of protein synthesis? Whole copy thing and
Water.
copy and, what's that?
The water side.
Yeah, how that led to protein synthesis. The lipid soluble hormone led to protein synthesis, yes. So I walked you through what the process of protein synthesis is, but let's back up and just talk about what protein synthesis really is. What is it? Protein synthesis is your body's ability to make or create or manufacture whatever it needs to build a new part that's worn out, or to create a secretion like a hormone that's going to be released throughout the body, whatever it is. So basically, whatever your body needs to make for itself to sustain life, it's primarily done through, and it's a protein structure. It's done through protein synthesis. Enzymes, hormones, molecule carriers, anything that anything that is a protein-based type of structure, anything that has a protein base to it or a backbone to it, is going to be done through protein synthesis. So what we see happening here is the DNA, as I described before, the DNA is like the master copy, the book. It's like the book that contains all the instructions on how to do whatever it is that you want to do. But it's trapped inside, just like a book would be at the library. You can't physically take the book out of the library. It has to stay in the library. So the only way that you can get the information on that page out of the library is to do what? Make a copy of it, either by handwriting it or by just making a Xerox copy. So that copy that you make that gives you the information you need is called the messenger RNA. The messenger RNA leaves, it can leave, and then it gets read by the ribosome. The ribosome reads out the instructions and puts together the protein that you need. It's going to assemble all the ingredients and make whatever the recipe is. If you look at the DNA as like a huge cookbook that tells you how to make any recipe imaginable. You make a copy of the recipe, you take it home, and then you become the ribosome. You assemble all the ingredients and you make it. And then whatever you make, the end result will be this little bead here of strings, which are protein. Because remember, a protein is made up of amino acids, right? That revelation to some of you. You all know that? Right, so the building blocks of proteins are amino acids, or you just nod because I said it. Now you know. So didn't know before. So we take amino acids, we put them together, make a protein. So the way that we do that, we go through 2 major processes: transcription, translation. Is this new, or have you been through this before? Been through it before? When?
In general, like,
You can talk me through this then, right? All right. So anyway, this is just showing you the making of the mRNA, and this is called the transcription. So transcription means to write, right? Correct? To write, correct? Or to make the copy. So this is the actual writing. So this is the actual creation of the copy. So that's one thing that I guess I don't even ask my students. I don't ask them if they've had it before. I need to teach it, and so ask if you've had it before. My job to teach it to you, even if it's again, right? Right. What's that? Yeah, correct. See? All right. So anyway, at least that's the way I feel. That I've heard. I won't go into that. On a record. Okay. Anyway. All right. So anyway, so what now happens during the process of transcription? What's happening is the messenger RNA is basically assembling the nucleotides to give you the template of how to make more of the instructions. So once you complete that, once you make the transcript, now it's going to travel outside and go to the ribosome. Now these are the genetic codes. These are all the different things that different combinations you could have. Adenine, uracil, or guanine, citrulline, right? Yeah. Cytosine, thank you. Oh man, supplement here. Okay, and you have some stop codons here. So these will be like the punctuation at the end of a sentence, and then this one is the start. It tells you when to begin as far as the translation. Okay, so when you think about translation, if I say, okay, I speak one language and you're going to translate it into another, you're basically reading back or you're reciting back something. So we have 3 sites on the ribosome. The ribosome is found in where in the cell? What part of the cytoplasm? You're correct. The rough plasmic reticulum, right? Sure, right? It is, yeah. So you think of like the pita with sesame seeds on it. There you go. Rough endoplasmic reticulum. The rough part of it is the ribosome. Ribosomes are rough, okay? So you get 2 parts of the ribosome. You got the top unit and you got the bottom unit, and they'll open up, or actually they're not assembled, and what they'll do is you'll take the messenger RNA and you'll lay it in there, and then these 2 subunits will close down on it and pinch it in between. So we have an active site, exit site, I can't remember the pairing site, maybe? I think it's pairing. Yeah, so active site, or active site, pairing site, and exit site. So what we go through in the order A, P, E. So you're thinking A helps you. Okay. So what happens is we start reading the codon, or the 3 nucleotide sequence. The start codon is AUG. So we know that we're starting to translate here. So you're gonna see that when we call out AUG, that's kind of like calling someone's name. Hey, I need Joey. And Joey's gonna come up, and he's gonna bring with him an amino acid. So Joey would be what we call the tRNA, the transfer RNA. It's transferring the amino acid to the ribosome for synthesis. Then after we are done with the P site, we move to the E site. Something else slides into the A site, so it'd be AUC. So I'm calling out someone else's name. So I say Jasmine. Jasmine's going to bring me up another amino acid. She's going to be the transfer RNA, and then we're going to stick the 2 amino acids together and keep doing that until you go. Following? Ring a bell? All this isn't earth-shattering or news for anyone, hopefully. All right, so this is what's happening. You're just building the chain. You get to the stop codon. Tells you to stop. You release the polypeptide, which is the protein, and you got your new protein. So that's the process. So we have transcription versus translation. The third term is the term transcription. Okay, so these pictures are just in the book and they're telling you, basically what I showed you, just giving you more about it. On Blackboard, you should have some videos. Want to brush up on your protein synthesis, or you want to see an animated version of it. There's some nice animations on Blackboard that I put there for you. Like a slowed down version, like step by step of the process of protein synthesis. And there's another one that shows you more in like real time, as you shows the thing zipping along and creating the protein as you go. So you can check out both of those. It's okay. So let's go back. So if you're doing mitosis, what are the different phases of the cell cycle? What do we start with? Before we do prophase, before we start peeing on the mat, what do we have to do first?
Synthesis.
Before synthesis. We have two G phases. So we have growth, the G1, so the cell grows a little bit. Then we go through, and then we go through growth 2. So G1, synthesis, G2, then we start prophase. Okay? Got it? So G1, synthesis, then we pee on the mat. Yeah. So prophase, what's the M? Metaphase, A is, anaphase, T is, telophase, okay? So metaphase is when the chromosomes are forming, things are condensing, it's getting ready to go. So we're kind of, and lining them up. So metaphase is get everything lined up. Anaphase is when you back up. Prophase is when the chromosomes are condensing. Okay? Prophase, chromosomes are condensing. Metaphase is when the chromosomes line up. Anaphase is when you pull them apart, and telophase is when you pinch it off and create the two cells. Okay? Meiosis. Oh, I'm sorry, go back. At the beginning of prophase, how many chromosomes do we have? 92. And then you produce how many daughter cells? Two with how many chromosomes? 46 each. And are they identical or non-identical? Mitosis? Mitosis. Identical. You got it. Okay, so then meiosis, you start with how many? 92. Then you divide into how many daughter cells? 2. This is meiosis 1. So how many chromosomes are in each of those? 46. Are they identical or non-identical? Non-identical. Then that's the end of meiosis 1. And then meiosis 2, then what happens? They divide into how many each? Yeah, so 46 will divide into 2. The other one, 46 will divide into 2. So how many potential daughter cells do you have then? 4 with how many chromosomes? Remember the sperm, that's pretty much how it goes. For the egg, it doesn't occur that way. You have the 92 divides off into 46 and 46. One of the 46 gets discarded. That's the first polar body. So now you're stuck with 46 chromosomes. Then after fertilization, or when fertilization needs to occur, then that 46 divides into 23. 23 gets discarded as the polar body, and then you're left with 23 to combine with a 23 from the sperm. Okay? So 75% of the chromosomes are discarded in the female. Meiosis. All right. So what would you say an allele is? How would you describe an allele? Yeah, kind of like the trait or the coding for a certain trait, right? Trait. Yeah. Sorry. Okay. So what are the first 22 pairs of homologous chromosomes called?
Autosome.
Oh, right. There you go. You'll never forget it now, right? And then what's the 23rd pair? Sex chromosomes. A male is what combination of sex chromosomes? XY, and then the female is XX. So with dominant recessive inheritance, tell me about dominant recessive. What's going to happen? What do you happen? Yeah. What about it? Happen.
You have dominant.
You have a dominant allele. Yes, it will always suppress the recessive. So if you have two dominant alleles, you'd say that's homozygous dominant. If you have two recessive alleles, that's zygous recessive. If you have one of each allele, heterozygous. You got it. Okay. Let's do incomplete dominance or codominance. Describe incomplete dominance or codominance? How does that work? The example of hair that we gave you. Traits of both and combines them. Yep. So if you have straight hair, one parent, curly hair of another, when they have children, their children probably have like wavy-ish type. Yeah, you got it. What about multiple allele inheritance? The blood for that example. Multiple allele inheritance.
Throw it against the wall and see what sticks.
Throw it against the wall and see what sticks. Oh, not that one.
Oh,
Is that the blood?
That's the blood one.
The blood.
Yep. Let's say I have type A blood. What are my alleles? IAI or IAIA, right? So you can have the same phenotype with different genotypes that lead to that phenotype, right? So a phenotype can be expressed through multiple allele combinations. That's what it means. What's a genotype? Your genetic makeup, your genetic coding, right? Yes. And what's the phenotype? What characteristics are shown, the physical expression of those traits? Exactly, yeah. You can have whatever genetics, the genotype, but the expression of that. So whatever, like your parents have a certain eye color, but you have your own eye color that's a physical expression of your genetic makeup. What about sex-linked inheritance? Color blindness. What does sex-linked inheritance mean? How does that? Some traits are only carried on the X chromosome. Why is it that they're only carried on the X chromosome? Yes, because the X chromosome can carry more genes because it's physically larger, has more real estate, more space. So there are some traits that are not carried on the Y chromosome. And so when does this become an issue? So like color blindness, when is this an issue? Mother carries the color blindness trait. Yes, that can be an issue. Her X chromosome to her son only has the option of carrying that chromosome with the color blind allele on it. Yes. Because the son is going to get the Y from the father. Obviously, that's what's going to allow him to be the male gender. So the only X that he is receiving is from his mom, and that has the color blind allele. But the male has no choice but to express that because there's no other X chromosome or there's no other color blind color vision allele on the Y to suppress that. Now, see, that's why you go through it again. You got it. That's why. Okay, and then what's the polygene inheritance? That's what you were talking about. Throw it to the wall, see what sticks.
multiple.
It was like the skin color example I gave, where you have parents, one has very dark skin, one has very light skin. The child will probably have intermediate skin color, but maybe not. Maybe they'll have very light or very dark, and it all has to do with your lineage, your genetics, going back generation to generation. Most of them are carried through that polygene inheritance. Most inheritance patterns are carried through that. I think that sums it all up. What questions do you have for me? Feel okay about the mitosis versus meiosis now? Go right up until the end, but we sure did today. Maxed it out. All right, thank you very much. See you next time.